Hello, everyone. Thanks for coming today. And today we're very excited. We have our own bioinformatician, Daniel McSkimming, Dr. McSkimming with us. Dr. McSkimming has a bachelor degree and a MA degree in mathematics from State University of New York at Buffalo, and has a PhD in bioinformatics from University of Georgia. And he actually just recently joined us not long ago, and my understanding is he's totally occupied. So, and he's the director of the bioinformatics core at the genomics group, right?
Mm-hmm.
And today he will talk to us. The title of his talk is Analyzing Microbial Composition. And thank you, Daniel. You have the floor.
Oh, thank you. So my talk is titled Analyzing Microbial Compositions, and of course we're going to be talking about microbiomes and how we query them and how we can gain information out of them. But I am going to talk more about a few different aspects of the experimental process. So here's a quick outline of what I'm planning on presenting. There have been quite a few talks in the last few weeks about how to analyze microbiome data and the types of statistics that we use, and motivation for why we would want to. So I'm going to try to move away from those topics because they've been covered, and to talk more about some limitations that we have with the different methods of detecting the microbiome, different ways that we can control our sequencing experiments and the benefits that those controls can yield for us. And then a very brief sales pitch. Hopefully, if there are any students involved, I would love to get some help developing some read processing pipelines, and I can talk about that very briefly. And then I'm going to talk about a little bit about analysis. And really, I'm going to focus on the central question that seems to be plaguing the microbiome analysis field right now, and that's whether or not the microbiome is compositional, whether or not it has a structure that we need to account for. Okay, I said again that I'm going to avoid the motivation. Why are we studying the microbiome? But I'll give a quick sentence. It seems as though the microbiome is involved with everything. By understanding human microbiomes, we can increase human health and human longevity, and we can start developing treatments and hopefully prevent a variety of different diseases. And the list of diseases associated with microbial dysbiosis seems to be ever expanding. But it's also important in agriculture for growth, development of agricultural products, bioremediation, and production of a lot of other substances as well, both medicinal and non. So it's a hugely important entity. Oh, I skipped, excuse me. So we have several methods for detecting the microbiome, detecting the composition of these organisms, mostly bacteria, that exist in these very niche environments. With humans, we have several different microbiomes. The most well-known is probably the gut microbiome, which we usually query with either fecal samples, or there are some interesting new devices that supposedly do time-lapsed sensing. It's a giant pill that you swallow that will sample your microbiome for you. But eventually we need to identify what bacteria are actually there so that we can begin talking about them, talking about the products that they create and how they interact with the host. Historically, the detection of different microbes has been through isolation and culturing, which we can see here on the right. This is a very time-consuming process. It tends to underestimate the diversity of the microbiome because we are going through this elaborate process of taking hundreds, possibly thousands of microbes and trying to piece out individual organisms. Now, there are also issues with being able to culture. Right? Current estimates say that about 99% of bacteria are unculturable. Now, that doesn't mean that they can't be cultured in isolation. It's more a statement of our lack of knowledge of the conditions that are necessary for that microbe to grow. So culture-dependent characterization is still hugely important. Right? We know it underestimates the diversity. We know we're missing a lot of organisms. But it really is the best way that we have to assess the phenotype of bacteria. And we can end up making slight changes to media and slight changes to growth conditions to understand how these bacteria work and how they interact with one another. Nowadays, though, we use DNA extraction methods, which started out almost as narrow-focused as the culture-dependent characterization, using sets of primers and qPCR-type assays to identify the abundance of just a handful of organisms. But then over time, they expanded into microarrays, and now we use sequencing. There are two main types of sequencing I'm sure everyone is aware of, both amplicon sequencing, where we're looking for a very small section of DNA from the microbes, and we use that in order to classify their taxonomy, and estimate their abundance. The other type of sequencing is shotgun, and that is more holistic in a way. We extract the DNA, put it through minimal processing, and then sequence it to see what's there. Now, each has its limitations and its benefits as well. So amplicon sequencing is dependent upon the choice of primers that we use. Right? We're amplifying a select region of the DNA, and so we have to specify an oligomer that says where the start and end of that region is. And we can see here, even in this example, the amplicons that were selected were using this red-yellow primer combination. And so we don't detect hypothetical bacteria number 6 in the amplicons data set. Right, that's a limitation of the primer. And nowadays, depending on what actual region you're sequencing, we actually look at primer sets instead of individual primers. Now, with shotgun sequencing, you don't have that issue, but it tends to be more costly. You have to sequence a lot deeper. The main technologies that we're currently using focus around the Illumina short read architecture, right, where we use amplicon sequencing, focusing on portions of the 16S, looking at hypervariable regions. So with the Illumina system, we have, again, short reads. Depending upon the chemistry, we can get up to maybe 300 base pair, paired end reads. And that's not long enough to actually sequence the full 16S region. So they tend to look at smaller hypervariable regions, and you'll see things like regions 1 through 3 or 3, 4 as commonly selected to try to identify the bacteria that are present. Now, of course, when we're looking at a smaller region, there's going to be less locations, less nucleotides that we can use to differentiate species. And so we end up having a poorer resolution of the taxonomy. Now, with shotgun sequencing, we, again, don't focus on an individual amplicon, so we can tend to get a higher taxonomic resolution, but it is more expensive. We end up having to have more reads specified or designated for each sample. So shallow shotgun sequencing is pretty popular right now. There's a couple of papers showing that shallow shotgun and deep shotgun both provide very similar abundances on diversity, or abundances and diversity estimates. And both shallow and deep shotgun allow you to estimate or identify which bacteria seem to be replicating. So there have been a couple of approaches that have come out in the last few years where we identify the origin of replication in these bacteria. And by looking at the ratio, the abundance of reads near the origin of replication, as opposed to the termination point for duplication, we can estimate the growth rate of bacteria as well. And so with shallow shotgun and deep shotgun, we can get away from the overestimation of the diversity of the microbiome by identifying which bacteria are dormant and not growing, not dividing, not participating in the metabolism. There are some long-read architectures as well, and I'm going to focus more on nanopore right now. Oxford Nanopore has recently released a kit for doing for amplifying the entire 16S gene and sequencing the 16S gene. Now, with the nanopore kit, they're actually looking at all about 1,400 bases, so they get a much higher taxonomic resolution, and they actually had much higher taxonomic resolution. Though I heard that there are some limitations on the primers. They're only using a single primer instead of a set of primers, and so there may be some bacteria that are missed. But interestingly, it seems as though the cost to perform a nanopore sequencing per sample may be on par with Illumina 16S sequencing. So it seems like it's both affordable and it's providing a higher resolution. A couple of studies have been performed that actually compare the nanopore sequencing to Illumina MiSeq, the short-read amplicon sequencing. And both of these papers have estimated about twice as many reads are actually identifiable down to a species-level taxonomy. So it seems as though by expanding from a small 3 to 400 base pair hypervariable region to the full 16S gene, we are able to increase the resolution of the taxonomies that we see. Okay, so that was a brief intro on the technologies that are out there. Anytime that we sequence any data, whether microbiome or otherwise, through one of these technologies, we want to be sure that we are including the appropriate controls. There have been plenty of studies, many studies, showing that the reagents that we use tend to have their own bacterial signatures. It's been termed the kitome, the microbiome of kits. And so whenever we're mixing any sort of buffer or larger volume of a liquid with our sample, we really want to have a blank of that buffer or that water that we're adding so that we can assess what the microbial contents of that buffer is, or that kit. You'd be surprised, even when things are certified DNA-free, there's oftentimes when we sequence it, we'll find bacterial DNA. Right? So by including these negative controls in our run, we can identify when there's contamination that may be affecting our participant samples and that could affect the outcome of the study. And so what I recommend is including a set of controls, both negative and positive, and then predefining cut points where you're going to say, if we have samples that don't meet these thresholds, we're not going to look at those samples. Right? Oftentimes we'll see thresholds dealing with the hit count, the number of reads that are assigned to a sample. And so here actually is the result of a sequencing analysis of a single plate that was run on a Illumina MiSeq. This was a 16S V3, V4 amplicon region of subgingival plaque samples. And we can see that we have both negative controls. We have extraction buffer and DNA-free water. And then we have our positive controls here, a mock DNA that people commonly talk about, and a pooled plaque sample that I'm going to go more in depth on in a few minutes. But importantly, we have a cut line here. Now, for this study, we actually had about 5 plates where we had run ahead of time so that we could examine the read counts and the hit rates and a variety of different statistics on the run and sort of identify where the optimal cut points would be. And so now we can see that with this particular plate, we end up having 3 participant samples where the number of reads that were assigned were too low to be considered a valid representation of the contents of the microbiome. One way that we can identify these optimal cutoffs is with something called the rarefaction curve. And we'll look at one of those in a little bit. But it is important to have these cutoff set defined ahead of time so that when you actually look at the result of a sequencing run, you can say, okay, these are the samples that are good, that are worth looking at, that are worth analyzing, and that are worth possibly publishing. Where the whole goal is to run statistical analyses and to identify correlations and patterns that lead to hopefully causality and then to publish. But if we're sequencing poorly defined and poorly represented microbiomes, then every step, every later step in the analysis is going to be biased. It's going to be affected by the contents. It's also important for large studies, especially larger longitudinal studies, to assess for technical drift. There's variation that comes into play with technicians, inaccuracies in measurement, machine changes in the machine, changes in lot numbers for reagents. All of these can end up affecting the contents of the microbiome that we assess. Now, for this study, this was a large study. I'll reference it a few times throughout the talk. So for a few details, we tracked about 1,000 postmenopausal women over 17 years. There were 3 different time points, and the main outcome that we were looking for is periodontitis related. Okay. Now, to assess the technical drift here, we actually created our own positive control, this plaque pooled sample that I highlighted earlier, where we took excess plaque sample from all of the participants, a large number of the participants, and mixed them together into a homogeneous mess. It was a very gross day in the lab. But what that allows us to do, we then aliquoted it out, put them each aliquot into -80, and as we run, as we sequence each plate, we pull out 3 of those aliquots and include them in the processing. Now, what that does is it tells us, just like the mock DNA, it gives us a measure of the success of our DNA extraction and subsequent sequencing processes. But unlike the mock DNA, the pooled plaque samples consist of exactly the bacteria that we want to detect, right? The bacteria that are in our participant samples. And so by using a pooled plaque sample, pooled plaque pool, I say it interchangeably sometimes. By using that sample, we can actually on each plate identify whether the bacteria that we're interested in studying are being extracted properly and are being identified. And one way that we can do that is with some statistical processes. So here I'm using, if we start at the bottom right, here's a graph of 30 different plates. So this study, I said, was about 1,000 women over 3 time points. We had some duplicates in there as well. And so we ended up having to run 30 MiSeq runs in order to sequence them all. Now, what's not included in this 30 are the 5 that we ran ahead of time in order to identify our QC cut points. And then also, we had enough sample failures where we would have to rerun plates or run new plates with those samples. And we also had entire plates fail, which happens sometimes. And so we ended up having to run a total of 42 plates in order to complete this study. 5 in the front, 30 that ended up being the data set that we analyzed, but then there were another 7 plates that we had to either rerun because of an entire plate failure or where we aggregated samples, individual samples that had failed previously. Now, this atypicality index tells us, we can see down here along the bottom in green, we have the pooled plaque samples. And this actually tells us whether or not the composition of the microbiome in those samples is consistent. Right? So it's not looking just at read counts or hit counts or any of these other sample level statistics. It's actually comparing it to the pooled plaque samples from our first 5 intro runs where we set up our QC. And it also shows us that the participant samples, which we see in red here, are all very different. The cut point is this 95th percentile, and that is actually almost a probabilistic-like score with this atypicality index. Now, we can see, particularly in the beginning, we did have a couple of individual pooled plaque samples that failed. They passed on the hit count. They passed all of our individual sample quality controls, but the composition wasn't repeated. There was a change in the bacterial composition that was inconsistent with what we would expect to see. And that's actually why we included 3. We wanted to allow that point of failure. So by having 1 fail, we would still consider the plate to be successful. Now, these types of controls can lead to additional benefits as well. So in the upper right, you can see that there's a figure that looks very similar, and it's essentially the same figure, except plate 15 looks odd. We can see that there are 3 samples that are labeled as subgingival plaque that look just like a pooled plaque sample. They're below that 95th percentile, so the composition is the same as a pooled sample. And then we have 3 samples up here that are labeled as pooled plaque but look exactly like participant samples. Well, it took a little bit of figuring out and looking at plate mappings and the like, but what we ended up identifying was each of these samples, each of these runs was processed in a 96-well plate. It means that there are 8 rows and 12 columns. All 3 of those plaque pooled samples, all of our samples were randomly placed, but all 3 of those pooled plaque samples ended up in the same row. And what happened was the technician accidentally, we assume, inverted the pipetting. So instead of going from left to right, when she was loading the samples, she went from right to left. And that ended up causing this error here. We were mislabeling our pooled plaque samples, which is what I detected with the atypicality index. But what that also means is that there were 9 other samples, participant samples, whose clinical values, clinical data, and metadata would all have been improperly attributed, which would have added noise to the experiment, added noise to the statistics, and made things more difficult to detect in the long run. So we were able to actually validate the remaining 7 rows of that plate using some negative controls, and there were 4 or 5 replicate samples. But it's very important to include controls, include known quantities in your sequencing run so that you can validate the data that comes out. When it doesn't happen, we can end up with a situation like this, where we end up with a set of plates over here on the right that all overlap. They look like what we would expect when we're randomly sampling from a population. But instead, on the left here, we can see that there are 3 plates that are tightly grouped together. And these all end up being a single phylum that's being identified. And what's more striking is that we can see that almost over a third of the variance in this data set is coming just from the differences in the sequencing runs. Right? Without having a control, like a pooled plaque sample, to assess whether or not there's a drift, these plates could easily be included in an analysis that would later, possibly need to be retracted if someone took a closer look at the raw data. So it's important to include controls, but it's also important to consider what samples you place on which plates, particularly with plates with long studies, longitudinal studies, or studies that have a lot of sample. And so I always recommend that if you have a variable of interest or covariates of interest that you need to take into account, that you uniformly distribute them across the plate, across all of your plates. That way, if there is a contamination that affects a subset of your plates, it's deconvoluted from the variable that you're going to be running statistics to. You're not going to identify that difference as being associated with the biological issue that you're looking at. I also think that it's beneficial if you're looking at a longitudinal study or repeated sampling to include all of the samples for an individual participant on the same plate. Now, I know that sounds almost counterintuitive. I just said we should evenly distribute everything across all of the plates. But if you keep all of the samples from an individual on the same plate, then that's going to minimize the variation within individuals, right? Because any contamination that affects them is going to affect all of the samples together. And when we actually are careful and uniformly distribute our variables of interest and keep our samples together, it can help us puzzle out issues, complications that will arise, that always arise. And so here we can see for one study, again, the subgingival plaque, about 1,000 women, 3 visits over 17 years. We can see that there's about 2/3, a little over half, 2/3 of the women on the second visit have this strange increase in M. flavescens, which is sometimes found in people, but it's a pathogen. It certainly wouldn't be found in a high abundance in an individual, at least not one that wasn't not a healthy individual. And what we were able to do, because we uniformly distribute our variables of interest, because we kept for each woman her first, second, and third visits all on the same plate, we could clearly state that this contamination was not related to the sequencing, It was not related to the periodontal status and to other variables that we were looking at, that it was only related to the visit. And we narrowed it as best as we could to a contamination, a reagent contamination when we were collecting on the second visit. But unfortunately, that visit was over a decade ago, and so we don't have any blanks of the reagents to actually verify that. But that was our final assessment. But when we are able to make those types of assessments and those types of identifications, then we can also do some interesting things to clean the data. And so here we were able to apply a Gaussian mixture model to separate out the contaminant from the non-contaminated samples. And then I used a KNN, a k-means imputation, to impute what a reasonable value, a reasonable abundance for M. flavescens would be in those samples. Now, this type of contamination removal and imputation processes, they do happen a lot. They can be helpful if you're going to end up modeling the data later on. But you have to be very careful. I wouldn't recommend reporting statistics as primary evidence on imputed data, because we would be artificially inflating the significance and artificially decreasing the p-value. So we can do things like this when the experiment is controlled properly, but we also have to be careful about how we use that data after imputation. Okay, that's all that I wanted to say about controlling their experiments. And so hopefully there are other benefits to controlling, and if people want to discuss that offline, definitely give me a call or shoot me an email. But right now I want to talk a little bit about pipelines and processing reads. Now, there are lots of great read processing pipelines out there. For 16S, especially Illumina-based, there's the DADA2 pipeline that works really well. And I actually have the image on the right is actually a representation of the DADA2 pipeline. But then Chime also has a deblurring pipeline. People have used minimum entropy decomposition. Lots of really great pipelines out there. What I'm interested mostly is not describing which pipelines you should use. There's pros and cons to each, and we can discuss that, again, offline. But I want to actually create a set of pipelines that are publicly available, certainly available to all the faculty at USF, so that it makes it easier for them to perform and process their reads on supercomputers, on HPC environments like research computing provides. Thus far, it's been slow going. So I have a couple of pipelines developed. I'm using a Python package called SnakeMake, which lets you chain together different pieces of software and different computational steps into a framework, so that when one step finishes, the next step starts automatically. And it also interfaces very well with the type of queue, the Slurm queue that research computing uses. Now I'm testing all of these on the HIPAA compliant RRA cluster, but we can definitely make copies of these pipelines and put them onto Cersei if people would like to use them on non-HIPAA type data. Currently, I have the Dada 2 pipeline and then a set of quality control pipelines developed. And I would love help if there's any students out there, if you're interested in bioinformatics or read processing or computer science and pipelining, please shoot me an email and we can set up some things on Cersei or get you access to RRA so that you can start contributing. Now, these pipelines themselves are actually, I'm using a version control system, so I have them stored on Bitbucket in a Git repository, which is just a way that we can have many people edit and contribute to improving these pipelines at the same time. So anyone that wants to get involved, please let me know and please come help because I definitely need that. In particular, I'm interested in adding testing so that we can automate some of the testing of these processes and documentation to make it easier for people to use them. More long-term goals, I think we could end up having some analysis type pipelines that automatically generate reports as well. But that is for the future, for sure. Okay. Once we process the data, right, for the microbiome, we're always looking at a table. And the table is going to be usually across the rows, we have our samples. Across the columns, we have taxonomy, right, units of taxonomy. I always tend to stick with that OTU framework, but that has some implications that aren't necessarily current. So nowadays, lots of people look at ASVs, amplicon sequencing.
They are the one who are benefiting now.
It's
a lot.
Is that a question?
You can get RNA isolation kit.
If anyone has questions, please speak up and let me know. I'm great answering questions anytime. And so we end up with these tables. If we're doing an ASV pipeline, like the Dada 2 pipeline, these tables can be very, very large. We can easily identify thousands, if not tens of thousands, of amplicon sequencing variants. And so what I always recommend is to aggregate by taxonomy. Now, some people will say the opposite. That's fine. Everyone has their own preferences. The reasons why I like aggregating by taxonomy is because it decreases the dimensionality of the data significantly. Right? Those thousands or tens of thousands of amplicon sequencing variants may reduce and often reduce to just a handful, a couple hundred of bacterial species. And oftentimes not even species, but a higher level classification, so bacterial taxa. By doing so, by decreasing that dimensionality, often orders of magnitude, we're increasing the statistical power. Oftentimes we end up running univariate statistical tests on each taxonomic unit, each taxon individually. When we do that, we have to account for the fact that we're running hundreds or thousands or tens of thousands of statistical tests. And we do that by something like the Bonferroni correction or the Benjamini-Hochberg correction, where we use things like family-wise error rates and false discovery rates to try to adjust our p-values to account for the fact that we're making so many tests, that we're making so many comparisons. So for both of those reasons, I think that dropping to a taxon unit is almost always the way to go. Now, an additional reason why is because I think it's very difficult when we do find an ASV that is differentially abundant. Let's say we're in a case control type study. Most of the time, I don't know how to interpret that. Right? If we have one ASV that's narrowed down to a bacterial species, that's only going to be a realistic situation in about 11% of bacterial species, because most bacteria have more than one 16S copy, or a copy of the 16S gene. Now, each of these copies tends to be, especially in the hypervariable regions, drastically different. They would classify as completely separate amplicon sequencing variants. So if we were actually in a situation where there was a bacterial difference, a change in abundance between our cases and controls, then we should be able to detect an ASV for every 16S copy number that that bacteria has, if it would be picked up by our primer sequences. I always find it difficult to interpret what a single ASV being differentially abundant really means, because it doesn't often impact an actual abundance of bacteria, change in abundance of bacteria. The other reason, another reason based off the 16S copy number, is that we can end up having an additive effect. So if a bacteria has 3 copies of the 16S, and each of those is partitioned into a different amplicon sequencing variant, there may be a small change in abundance between cases and controls, but by adding all 3 of those together, we would actually triple the difference in read counts, or difference in abundance estimates, which would give us added power to detect the difference. So it always seems like it's a benefit to me. But when we do, and so yes, let's take a look here. We should also consider strain variation, but that only makes sense when we already have a species level taxonomy assigned. If the taxonomy isn't at the species level, then we know that there's going to be multiple species in there, not just multiple strains of the same species. Okay. Once we've done this, either aggregating by taxonomy or not, we can start looking at things like rarefaction curves, which is how we can set cut points for significance. So in the beginning, we can see the slope here is very high. That means a small increase in hit count, a small increase in the number of reads leads to many new bacteria being discovered. And so what we tend to do is try to find a point where that levels off. Now, we're always going to identify more bacteria with more reads, new species, even if they're coming out of possibly just from sequencing error. They're not necessarily novel species themselves. But we do have to make a decision at some point. And so once the slope levels out or becomes less than 1, we can start saying that we're representing the composition of the microbiome. Before that, we would only really be representing a random portion of it. And then we can also, of course, do our normal alpha diversity type assessments, where we're looking at the, in this case, the richness, because it's a Cal1 score, of the OTUs, the number of OTUs that each sample has, to see if there's an increase in the number of bacteria. And here we do see that there is a statistically significant increase using ANOVA. So it's a shift in means. And that we expect with periodontitis. It's a well-known phenomenon. Okay, but we can also, like I said, detect strain level differences. Here I used an algorithm called minimum entropy decomposition. But again, no matter what algorithm we use, when we're looking at amplicons, when we're looking at small regions of a sequence, of a species, excuse me, we need to keep in mind that there's going to be multiple copy numbers. Right? And so here, this L mirabilis, we can see that there really isn't that much of a change across the, as we increase our degree of periodontitis. And so we wouldn't say that there are strain level differences, but L mirabilis has a copy number above 1. And so many of these, this is probably 1 or 2 species with multiple ASVs identified. Similarly with Tannerella forsythia, right? There's a dominant strain or a dominant ASV that we identified, but then many, many others that are either low abundance or small strains, or could be just sequencing error at that point. But we can see in several periodontal pathogens like T. sacransky that there is a shift, and those individuals with a higher level of periodontitis, which would be denoted by this 3 here, tend to have one version of T. Safransky, or one strain of T. Safransky, whereas those who have none or mild periodontitis have more of a different strain. Okay, but all of these questions lead us, or all of these observations really lead us to the primary question, because we're going to want to be looking for differential abundances, detecting differential abundances, looking for correlations between bacterial species. That's how we identify things like interactions within the microbiome, or one way that we identify interactions. And we'll want to do things like generate regression models and machine learning models. And all of these really necessitate us answering the question, is the microbiome compositional? Because the approaches change. And so here we can see with a standard approach, we have one set of tools that we'll go through. The compositional approach, we have a different set of tools. Now, this concept of compositional data sets came about, or was first published by Carl Pearson in 1897. He was working with a biologist, Weldon, and Weldon was dissecting shrimp and weighing and measuring the shrimp in different ways. And they ended up having a dialogue back and forth where Pearson warned about spurious correlations that can arise in the data when he called it using, when you use a common index. Since then, it's been expanded into this framework called compositional data that applies whenever we're looking at parts of a whole. And the idea with this parts of a whole is that it doesn't matter how many reads necessarily you have representing your whole. There's a minimum threshold. Once you get above that, the important part isn't actually the number of reads. And if you think from a fecal sample, if you take one sample and sequence it, you would get a certain amount. But if you multiplexed it with 100 other reads, or 100 other samples, you would get many, many fewer reads that are still describing the same entity, the same composition. And so what the main observation that came out that really allows these compositional approaches to be developed is that the information in the data set is not the counts of the reads themselves, but the ratios between counts. Okay? It's a lot. We don't need to actually think about those ratios a lot, although you will hear the term log ratio for the next 10 minutes or so. Now, what this compositional framework says is that when we have this parts of a whole, there's a sum constraint that is going to be forced onto it, either by the total number of reads that are assigned to a sample, or if we normalize that, often by dividing by that number, it will be constrained by 1. We wouldn't be able to have any that are greater than 1. But what it actually ends up being is a completely different domain that the data lives in, called a simplex. We don't need to get in depth about that, but I'll mention a little bit more about the simplex and ratios in a bit. Now, the main techniques are very similar. We have a set of operations that we want to perform on our data. The way that we normalize is really the biggest difference. And so common approaches for normalization, rarefaction was an approach that's commonly used, hopefully not still. Several people have written very critical articles about rarefaction. But then there are other packages out there like DSeq and EdgeR that have normalizations that are things like TMM, trimmed M means. Approaches that attempt to normalize the number of reads that are assigned to each sample, right, is really the framework for this normalization of the standard approach. Whereas with the compositional approach, we're instead looking at ratios of reads and then taking a logarithm. Distances can be a little tricky. The preferred distance in the compositional approach is called the Aitchison distance, but it's very simple. It's our common Euclidean distance on CLR transform data. And so we're basically, after we transform we're basically using a Euclidean distance in many cases. With the standard approach, we have some other issues, though. Well, some of these are distances, like unifrac and the Jensen-Shannon. Well, the Jensen-Shannon is a divergence measure, but if you square it, it becomes a distance. But measures like Bray-Curtis aren't actually distance metrics. And so that can lead to some issues later on. They don't, there's three different principles that a distance needs to have, a distance measure needs to have in order to be a metric. One of them is called the triangle inequality, and Bray-Curtis fails it. For ordination, there's not a whole lot of difference between the two of these. I prefer PCA because we are looking at variation within the data set, and with PCOA, you're actually looking at distances and preserving distances. But we can see, and this I think is the most important part, for multivariate comparisons, it's identical. Right? They're saying that our initial assessment of beta diversity, whether or not there's a difference in the composition of a microbiome between cases and controls, it doesn't matter which approach we use. And this actually ties back into Pearson's 1897 paper, because the recommendation that he gave Weldon in order to accurately and efficiently analyze his data set was to use a permutation-based test. And he described a way of randomly generating a background distribution for comparison. And that is exactly what we do in both Permanova and in OSIM, which is why they're the same for both. It's an approach that will work in either case. Correlations fall apart. Again, Pearson himself warned that Pearson correlation wouldn't work. And we have some tools for differential abundance, although each of these has limitations. ALDEX 2 recommends you have at least 10 samples per group that you're comparing. ANCOM can be overly conservative. They actually run, if you have, let's say, 100 organisms, they're going to run an order of 10,000 hypothesis tests and the like. What I think is more interesting, though, is moving away from differential abundance and moving into things like regression analysis and machine learning techniques. And I'll get to that very quickly. Why I say that the microbiome is compositional is for two reasons. The correlation of relative abundances does not capture the correlations in the raw data. And here's a toy example that was published in 2016. If we take this data set here, pretend that this is 3 samples of, let's say, 3 individuals, and we're measuring 4 different bacteria, then we can see in the upper right corner here, we have one set of correlations in the raw data. But if we calculate the relative abundances by doing a read count type normalization, we end up with the correlations in the bottom. And there are some very drastic differences, including some that go from completely, almost perfectly negative to a much weaker negative, and some that go from a positive correlation to a negative correlation. Okay? It's actually a little bit more complicated. In this framework of the simplex, we actually have an array that represents each point. So here we have an example of 3 different data sets of absolute abundances, and this is a toy example, of 2 different messenger RNA, and a collection of 7 different samples. Right? In this first absolute abundance, we have almost a perfectly negative correlation between the samples, or between the mRNA1 and mRNA2. In the absolute set 2, we have an almost perfectly positive correlation. In the absolute set 3, we have almost a 0 correlation. All of these data sets, if we take a relative abundance, reduce down to this red set of dots here, which again has its own correlation. So in essence, when we're using this type of read depth normalization, we're choosing where along these rays we want the point to be based off of the read depth. And by doing so, we're choosing the set of spurious correlations that we're then examining in our statistical analyses later. I'm completely out of time, so I'm going to go very quickly. John Aitchison worked out a lot of this compositional type analysis, or at least one main framework for it, which involves taking a log ratio transformation And by doing so, you're removing that sum constraint. You're transforming the data out of that simplex and into an n-dimensional real space where it has a well-defined covariance structure, and we don't have these correlations that change based on the read depth. And then he recommends just applying standard techniques at that point, often assuming a normal distribution in the background. There is a second approach that can be used as well, where we consider the operations in the simplex. Addition and multiplication don't exist in the simplex. They're replaced with operations called perturbation and powering. I forgot the second one for a second. But we can also redevelop statistical tests for so that they can be run in the simplex. Both approaches have many limitations. Zero inflation is a huge problem with compositional data, particularly when there's lots of different samples of varying diversity. And the standard approaches, there have been several reports that false discovery rates aren't being controlled properly. But I'm a pictorial kind of person, and so on the right here is what I see when I look at a standard data set. Now here I looked at, I believe, relative abundances. So I did a read count normalization. And it doesn't look like a data set that we can interpret meaningfully. We have these 4 vectors of points coming out, 4 lines. And most of the variation in the data set doesn't make sense. It's not associated with, it seems to be associated with differences in the infant microbiome. So green here is an infant preterm microbiome. Whereas if we apply a compositional transformation, then we come up with a picture, a view of the graph of the data that's much more meaningful to me. It looks like something that I can run statistics on. It looks like these infant samples may cluster into several different clusters. And we can see that the variation in the data set now becomes biologically meaningful, where we're separating out these low diversity infant microbiomes from higher diversity, more fully developed adult and preschool toddler microbiomes. So it seems as though the data set, even in a very superficial level, is more interpretable. And so that's why I lean towards compositional techniques. But I'm happy to discuss any type of technique with anyone. So for future directions, I see 2 approaches, or 2 things that I would really like to work on. And one, I think, is incorporating this idea of zero inflation with compositional data analysis. The transformations that we use make that very, very difficult. And so I think that there's some creativity that might need to be exhibited in order to really answer that question. And the biggest problem that I see is, again, when we're mixing, trying to analyze in a single framework, samples with both low and high biomass. For example, infants who might have 2 or 3 bacteria in their entire microbiome, they're less than a week old, with adults that have hundreds of bacteria. I also think that we need to start pushing some deep learning approaches. There's 1 or 2 that have been published, but there's a lot of room for growth there. And the biggest benefit that I see when I think about combining these neural networks into layers is that right now, even with classical machine learning approaches, we are manually constructing data representations. Right? When I take a log ratio transformation, I'm manually constructing a new representation of the data and saying that it's more meaningful in some way. But with the different algorithms with backpropagation, we actually end up having the nodes themselves in these deep learning approaches decide what the appropriate representation is, and can look at many more representations than we realistically can. I went a little bit long. I'd like to put up a quick list of acknowledgements. A lot of the work that we saw today was from University of Buffalo, where I work with some epidemiologists and some dentists. And then a good portion of it was also from work from here at the University of South Florida. Thank you kindly.
Thank you, Daniel, for a wonderful talk. We have a few minutes for some questions. Anyone from the audience have any question for Daniel? Okay, Perika Herman, you raise your hand. You are on mute. Dr. Herman, do you have a question? Oh, Dr. John Reyes, do you have a question?
Oh, I can ask them. Thank you for giving a very nice and technical talk, Daniel.
Well, thank you. You're welcome. It was my pleasure.
You're welcome. USF. So you said you made the recommendations that to reduce the dimensionality, you always consolidate by taxonomy before you go further with it, right?
I do. For several reasons. Is there a specific question, or I keep cutting you off? I'm sorry.
Yes, I do the same. I do the same because I like it for all the reasons you mentioned. And if you reduce the dimensionality, you can apply a lot of analysis, otherwise it would not be possible. However, I always have a dread in the back of my mind. That's what I'm trying to ask you. Because I'm afraid we missed something essential. The reason why is the classification of the microbiology in terms of microbiome is incomplete. So we learned the lesson from IBD in gut microbiome. So if we collapse all the data by taxonomy, actually we will misclassify one group of bacteria, because so that one is the Baudia. So the thing is that there is a finer gradation and classification in that official taxonomy that make people actually quite sick. But the knowledge is incomplete by the time if I collapse it, I lose it. It's only in retrospect. So I don't quite know what is the real solution for it. That's
I imagine, and that definitely is a situation that we can run into. I imagine that in that situation, if we first aggregated by taxonomy, right, those reads would all fall into a species level categorization. And so then we can go and look at beta diversity differences limited to the ASVs within a specific taxa, a specific species identification, right? And in that way, we're actually performing that subspecies or strain level type analysis. There are lots of issues with aggregating by taxonomy as well. I mean, it seems as though no matter what we do, there's always a downside. And I think another issue is that a lot of these ASVs don't actually have a taxonomy that's applied to them. Many come up as not recognized or not identifiable. And so we would, again, need to go back, and the only thing that we could really do with that type of sample as well would be to look at the ASVs. But I think that as a first pass, going to a higher level taxonomy is usually a benefit.
Thank you. We have time for another question. Any question from the audience? Yeah, I have a question, Daniel. Could you share more your thoughts about how to use deep learning to find the better representation for the microbiome data?
Certainly. So for deep learning, from my understanding, we would end up taking a variety of networks of neural networks and combining them, stacking them in different ways. And there are lots of, in some ways, to me, my understanding of deep learning is very minimal at this point. I'm just getting into building these type of models. But it seems as though a lot of the novelty that comes into these deep learning networks is really in how the networks are connected and how the nodes are connected. And so I would think that we would end up having to have, at least in the initial point, have very dense connections so that you could end up really evaluating whether or not things are varying, how things vary with one another.
Right. But I mean, the deep learning is really good at analyzing like images, right? Right. And is the raw microbiome data actually was an image?
You can think of it that way, right? You can think of it as a heat map, that table that you generate. And that is one of the approaches that has been used, is people have actually gone and used a convolutional type neural network, which is used more in imagery analysis, and created heat maps and fed heat maps through a convolutional neural network.
Did they find anything?
They posited as I haven't read anything since. That was at a conference that I was at in 2019.
I see.
And I haven't read anything about it since. So it hasn't.
That's a very interesting perspective. I asked Dr. John Adams, is that a good approach? He said no. I didn't understand why.
Well, it's until we actually get more of these approaches out there and can evaluate one against the other, I think it's going to be difficult to really say, what is the best. But they were able to do some. They were able to find some meaningful associations in their studies. So.
Interesting. All right. Last question, anyone?
Looks like there's a couple of questions in the chat.
Oh, okay. I'm sorry.
Yeah.
Let's see. Okay. Okay, there's a question from Dr. Parikh Herman. It says, do you use any method for the batch correction between different batches?
Yes, batch correction is always a fun thing. I try not to use batch correction if at all possible, because it is in some sense it's a lot like imputation, right, where you're making an assessment that something is a contaminant and then removing it from your data set. And so I like to stick with the data as natural as possible. But in some situations, we can identify those batch issues. If it's very strongly associated with a batch and it's limited in scope to a handful of bacteria, then I think it might be worthwhile applying a batch correction or doing a process like I had done, where you use like a Gaussian mixture model to separate out the batch effect and then re-impute a new value. It always makes me feel like we are fiddling a little bit with the statistics when we do a batch correction, and that we're going to make things seem more statistically significant than it may be. So again, you have to be very, very strongly assured that you're looking at a batch, a batch effect. And even then, I would make sure that the data that you pass to these batch correction, so there's combat, I think is one that's popular in R. I would make sure that you pass just the data that you need in order for that batch to be corrected. You don't want to have them adjusting data that seems to be that seems fine.
Okay. I hope that answered your question, Dr. Herman. There's another question from Dr. Yan Xiao Li. Could you comment on recent paper by Calgaro, 2020 assessment of statistical methods from single cell, bulk RNA-seq, and metagenomic applied to microbiome data?
Yeah, I'm searching for it right now. I'm not sure of that paper by the title. That's something I would have to read through this paper. I'm not familiar with it offhand.
Right.
So if that's something, Dr. Hansen, if you'd like to shoot me an email,
Yeah.
that's something that we, or I'm sorry, that was Dr. Lee.
Dr. Lee, yeah, yeah. Okay.
That's something we talk about
Maybe
offline.
we can talk this about the last question from Dr. Barbara Hansen. She said, appreciate your emphasis in the early part of your lecture on the importance of a control group. Do you have concern about the two having very different variances?
No. I think that, so I consider variances to be within the groups that I'm considering usually, like within group and between group. And so if I'm negative controls, we would expect really to have very ideally no bacteria identified or very few. And so they would naturally, I think, have a slightly lower variance than a participant sample that would be a much more populated. But that is something that I'll have to think about, whether the effect of the variance, changes in variance would have there.
All right. I hope that answers Dr. Barbara's question. Okay, I think we're 6 minutes over time. Let's thank Daniel for a wonderful webinar. Thank you, Daniel. And thanks everybody for coming today.
Thank you for having me.
Yeah.
Thank you.
Thank you. Take care.